Thank you. Thanks a lot. Let me talk about the new adventure I'm on. I was at Caltech for 27 years, very happy, and where I also did this acceleration with neurosurgeons recording from individual neurons in cancer patients. But subsequently, I left Caltech to dedicate myself to a new 10-year mission that I'll tell you about. And we need help, particularly from information theory. I work with quite a few of them. So let me first talk about cortex. So cortex in us is roughly, it's like a pizza, so it's actually like two pizzas. So cortex itself is a layered structure. It's a 2 plus epsilon dimensional structure, meaning if you look at across all mammals, and cortex is stereotypical for mammals, neocortex, its width only changes by factor 2 to 3, let's say from a mouse where it's 999 micrometers to a human where, or an elephant where it's 3 millimeters, but it changes by a factor of 50,000 in extent. So in us it's pretty much like this. It's 1200 square centimeters, pretty much this thickness, and of course it's folded together because evolution had the challenge to pack two of these cortical sheets into one skull. So think of it as a computational tissue, 2 plus epsilon dimensional computational tissue whose extent varies dramatically across different species according to the ecological niche. It is relatively uniform. So yes, there are differences between one area and the other, but basically, I'll show you some pictures. To a vast extent, it looks like the same stuff everywhere. Whether you're looking at visual cortex or in motor cortex or in prefrontal cortex, basically it's all the same. It's a vertical organization. There's a whole bunch of neurons that are organized this way. The neurons are similar, albeit not identical. And same thing, if you take a little piece of mouse tissue or you take a little piece of monkey human tissue, only an expert armed with a microscope can tell one from the other. So the null hypothesis that we are exploring that the main difference between us and the fact that we can sit here and give talks about information theory, and the fact that the critters that I'm no doubt of live here somewhere in the basement, okay, mice, the fact that they can speak is that they only have roughly 1,000 times fewer cortex, so between 1,000 and 2,000 depending what method you're dog. And the null hypothesis is that the basic hardware of the brain is very similar, not identical, but it's very similar across all species. What really varies is the architecture. And of course, if you have 1,000 times more, you can have many more levels of hierarchy, many more levels of abstraction. And we all know from deep learning from convolutional nets how important multi-layer architectures are. Of course, there are myriad of differences between the brains of all different species. The question is there any one dramatic difference or the magic bullet that people always look for? We can't exclude it. There may well be something that allows us levels of computational performance or levels of abstraction that a mouse is not capable of, but so far we don't have the evidence for that. And so the mission that we, in particular at the Institute, I'll tell you about us, and in general people have right now, particularly under the BRAIN initiative, is trying to figure out what is the secret of natural intelligence. We know natural intelligence is most closely linked to cortex. It's not the only structure that's involved, but it's the key that's involved. And so people are looking, what is if you take a unit, a unit piece, a so-called column of this structure, what is the nature of the computation that's being performed that allows it to be replicated both within the sheet and then in many, many layers of a hierarchy. So here you have a well-known model, Tommy Poggio, my advisor at MIT, the model that he and his collaborators built over the last 20 years of visual object recognition perception. This is a model we have based on monkey data, starting in the retina and intermediate way station primary visual cortex, which is at the back of your head. You can feel it, there's a bump, it's close to where your primary visual cortex is. And then in the monkey, it goes to a plethora of visual areas, roughly 36. In us, it's roughly the same. And so what people have done since 40 years, starting with tubal and basal and neocognitron, and then these sort of model by Poggio, and then of course Jeff Hinton, Jan Kuhn, etc. They've made these very simple models, essentially simple cortex, where you have one stage where essentially you do very simple computations with a very simple nonlinearity. A computation, all you need is a half rectification followed by a pooling operation. And there are various ways you can pool. You can do an H-max, or you can do linear pooling, all sorts of ways. And then you repeat this linear nonlinear step. You repeat that a number of times until you finally get to the stage where you have things like faces versus dots, and where you can also have things like neurons that respond to Jennifer Aniston or to anybody else who you're very familiar with. And of course, this here I stole a slide from Jan. So this is the same sort of architecture. There you have Schubel and Wiesel, who discovered the original one. And if you look under the hood, under these convolution nets, you find the same sort of operation. You find a nonlinearity, stationary nonlinearity, normalization, sometimes whitening, and then cooling. And that's replicated. And of course, we all know how well that works, how extremely well that works. Now, if we go further under the hood, if we look in any little bit of cortex, what we see, this is done from Callaway, who's here at the SOC. So this is a quarter of a millimeter. We see all these cells. This is a Nissl cell. And then sort of, if you do the detailed staining, what you see, so here he stained a couple of cells, you see these very complicated things called neurons that we know of since roughly 150 years. Now the challenge has been, or continues to be for neuroscience, that the brain, if we stick with our brain, consists of a very large number of these things, between 10 to the 8, 10 to 11, depending on which species you look at. They're highly heterogeneous. I'll talk about that. This is a big discovery that people made over 100 years ago that still are not at all reflected in theories, in computational theories about the brain. So there isn't just one cell type. A neuron is a neuron is a neuron, and all that matters is just the WIJs there. But it turns out just in the retina that you think, well, all that it does, it converts, it's just like a CCD camera. It converts photons into spikes. Turns out there are 80 different discrete cell types, each of which has a specific job. And the output of a retina is 20 different cameras that go to different parts of the brain, each one highly specialized to detecting motion in one direction or the other direction, that detects looming things, that detects the overall luminosity, that detects contrast changes. And where these different neurons compute different spectral, they cover different parts of the spectrum. So they interact. The fan out is also, the fan in and the fan out is very high. So in cortex, typically neuron may get input from between 1,000, 10,000 other neurons and project to 10 to the 3 to 10 to the 4 other neurons. And of course, the operation occur over a wide spectrum of spatial temporal scales, right? In us, the typical neuronal operation, something like generate an action potential, a particular type of pulsive nonlinearity that's involved in communicating the results from one neuron to the next, occurs on a millisecond scale. And of course, we can remember things over, a meeting that we both attended 10 years ago. So this is a huge challenge. The second challenge is we do not have a list of the cellular components. So there are roughly 100 billion neurons. They're probably of 1,000 different types, but we don't know exactly what the types are. And we don't know how they're interconnected. We know the rules are very specific. So think of a Lego set. Think of, you have 100 billion building blocks Lego set, and there are 1,000 different types. And it's really critical to figure out the fact that the 2x8 blue stones only fit on the 1x3 roof tiles, but they don't fit on the green 3x3 stones, right? And we need to figure out those rules, but we haven't figured that out because we don't know the different components. It's very difficult to simultaneously record from more than a tiny, tiny fraction. You can do one of two things. You can either record from the entire brain using brain imaging, then you can visualize the entire brain. That's pretty cool, but you do it at a very crude scale, at a very sedate scale, typically at second scale, and typically your minimal unit of resolution is several voxel. That'll include 1 million neurons. And of course, you're not looking at neural activity, you're looking at blood flow, at hemodynamic, at a proxy, at power consumption. Or you can do what we did and what many people do, you can put 1 or 100, and now people are building 1,000 wire electrodes in the brain where you can record from 1,000 neurons simultaneously out of 10 billion. So that's still a dilution of 10 to the 7, 10 to the 8. There are no accepted standards. So neuroscience is heavily balkanized, unlike some aspects of psychology or physics. There's simply no accepted standards. So when you think a spike is a spike is a spike, it turns out not to be true. Depending on where you got your PhD, with which advisor, which operating system they were using,
I'm dead serious. You're going to use different spike registration software. You're going to use different spike identification software. And if you put the data online, which the vast majority of labs, of course, are not doing, then you put, essentially you put a tarball of your files, of your data online, which is essentially totally incomprehensible. And even for such basic phenomena as spikes, what is an oscillation, synchrony, sharp wave, we don't have accepted definition. And that's the thing that I see. People are more likely to use each other's toothbrush than to use each other's spike detection software, because I'm incredibly comfortable with my advisor used it, her advisor already used it, and so I'll be damned if I'm going to use your thing. There are no central unifying projects, unlike for example, astronomy. And they are on the order of 10,000 labs, roughly. There are roughly 60,000, 70,000 neuroscientists worldwide. They're constituted in these 10,000. Think of them as 10,000 independent contractors, because essentially, that's essentially what we are as academics. So there's 10,000 contractors, and of course, nobody ever wants to be told by anybody else what they should do, right? That's a great thing about being an academic. As I said, I was an academic for 27 years. It's great because you can do anything, in science as long as you can get funding for it. So they pursue, how many of you have been to a neuroscience meeting, like the big one? Okay, so the typical meeting now of my community involves 30,000 people, where literally you have a mile-long poster sessions, okay? And if you walk past down this mile, I mean, it's great aerobic workout. If you walk past there, it's just dizzying. It's just amazing the variety of animals people record from, techniques they record from, different ages. Some are very young, some are juvenile, some are adults, some are older, different techniques, different technology, different methods. And so think of it like a sociological Big Bang, right? You have all these neuroscientists. We want to understand the space of how does the brain, how do nervous systems compute? And so everybody goes off on their own. In fact, the system, of course, is designed, it's by design built for orthogonalization, right? You have to be as independently as possible in order to get your PhD, in order to get your high impact paper, in order to get tenure, in order to get your grant, right? You have to do something as different as possible from everybody else. Otherwise, you're not going to get those rewards. And so that leads to the fact that everybody's off doing their own thing in general. Of course, you come together briefly for convenience, for example, because somebody dangles a grant in front of you if you work together. But in general, people are very, very independent, which is a great thing. It's a genius of the academic system in the modern world, but it also makes it very difficult, if not impossible, to do standardized science that's repeatable, where you can replicate what the data is outside, where everybody agrees there's certain things that we ought to do. And the dirty secret in biomedical science is, whether it's FMI or neurobiology or cancer biology, that 2 out of 3 experiments cannot be repeated. Or if they're repeated, they find different results. All right, and a lot of these, so people have done this, you take studies in Nature and Neuron and Cell, some of the top science, the top-line journals, okay, published, heavily refereed, people try to replicate them, for example, in industry, and typically they will fail. More than half the time they will fail to replicate the experiments. Why? Well, as I said, because trying to replicate, exactly your condition in the absence of any standards, if everybody does something different, is almost impossible. And of course, unlike physics, where you typically study systems with very small degrees of freedom, like electrons or black holes, etc. Here we study things, even a synapse, let alone a living animal, is highly variable with a very large number of degrees of freedom. So it makes it very difficult. All right, so universities are simply not set up for large-scale systematic efforts. For that, you have to go to national observatories, you go to CERN, or I'm going to claim, you can also do an alternative models like CERN or like LHC or like a SLAC, etc. That's what we're trying to set up for biology. And finally, there's limited interaction between experiments, modeling, and theory. Typically, because of the way you get tenure and things like that, you're typically consigned to your silo. All right, so we try to offer a different model. It's not a better model, it's just a different model. Universities, as I said, are fantastic. They're great in many things, particularly for discovery. They're essential. But as I said, I think they're not so good for doing things systematically. In particular with the brain challenge, I think, the one that President Barack Obama announced that people here, particularly at SOC and UCSD, are heavily involved in Ralph Greenspan, Terry Sejnowski, etc. It really calls for much, much tighter interactions than previously. So what are we? We are a non-for-profit medical research institution. We have started 12 years ago. We're growing rapidly. So when I came, we were 160. We're now 300 roughly, and we'll be 500 in a couple of years. So our culture is between a university and a biotech startup. So we are a little bit like a university in the sense that most of the scientists, we roughly have 100 PhDs now, they all come from either grad school or postdoc or faculty like me or some of my other colleagues from Harvard, Stanford, Caltech, MIT, etc., the usual places. But we are very different from a university in the sense that we have a few goals that we decide as a team together with outside advisors. We as a team, and we execute on those goals. We have milestones, we have so-called SMART goals, we have deliverables, we have program management. So in that sense, we're quite different from a university. In that sense, we're much closer to biotech. We have products. I'll show you. We've had so far 33 successful product releases. The products are data releases or tools, and we give it all away. Everything we do, we give away for free. So I came to the Allen Institute, and then together with my CEO, Alan Jones, he and I proposed a 10-year plan to Paul Allen, in whose name, of course, and who started the entire institute. It's for 10 years, for $1 billion. And we're now in year 3. Well, we almost finished with year 3. We're now entering year 4 on this very ambitious plan. That's currently our budget. Most of it is made possible by the unprecedented generosity of Paul Allen. He's really an exceptional individual who spent already now more than $500 million on basic science. And just this year, he gave another $100 million for Ebola. He started just a couple of months ago another institute for cell sciences. He's very much interested in understanding how things work at a very basic level without trying to make any profit from it. As I said, we're completely non-profit and open. So that's Paul. That's last year. We almost made it again this year, but 1 yard to spare. And we're getting a new building. Paul is also giving us a new building just next to Amazon, to the Amazon headquarters. We're getting our own 270,000 square feet building just for brain science. So as I said, we've 3 times a year typically we have data releases that we release. So we started way back in almost 10 years ago with our first product, a large-scale atlas of all the genes in 20,000 mice. So if you look for a particular gene that you're interested in, let's say something in memory or in schizophrenia, whatever, you can go to our website, you can download a three-dimensional brain browser, and you can see in high resolution, in detail, where this gene is expressed in the mouse brain, in the developing mouse brain, in the developing monkey brain, in the monkey brain, in the developing human brain with fetuses, and in the human brain. Over the last 10 years, we've released all these atlases. So these are very large-scale resources that will take between 50 and 100 people to produce and to put online. Now, in the second phase of our operation, we decided we wanted, because Paul was challenged us, we should do more rather than just generating data and pushing it out there. We should generate our data ourselves and have much more research science in-house rather than just expecting in the community. So we're moving in that direction. So what we're going to release in a couple of months is our first product of those. One is, I'll talk about a very detailed description of neurons, their electrical properties, their morphological properties, and separate their transcriptional properties, as well as we're releasing a new high-resolution three-dimensional brain atlas that has 500 million voxels with a resolution of 10 by 10 by 10 micrometer. Right? The typical brain atlas in the human, of course, is a millimeter. This is 10 micrometer because, of course, we can do things in a mouse that you can't do in a human. Roughly half of our resources are human-based neuroscience, all cellular-based. I won't talk about that today, really, with one exception, and the other half goes towards the mouse. So we have a lot of data. Just for the mouse, it's 3 petabytes, more than a million slides, all online. 50,000 unique visitors. We make a lot of transgenic animals. Of course, one reason why we can do this, 2 reasons why we can do this today, this incredible detailed exploration. One is because we can build transgenic animals in a mouse where you label, for example, a very specific type of neuron. Let's say only one type of neuron in the retina that only fires to upward motion. Or one neuron in cortex, one type of neuron in cortex that inhibits one particular neuron. And furthermore, through the power of optogenetics, we can now then also insert tools, molecules inside those neurons to turn them on or off with millisecond precision at our will. So we can now intervene at a point in time of our choosing in the complicated networks of the brain very delicately, very deliberately, and so we can move from observing brain activity, just seeing, for example, every time the patient looks at a picture of Jen Anderson, this neuron fires, to intervening. What happens if I remove that neurons or those neurons that fire to Jennifer Aniston? Will the patient then be unable to say, who? Well, I know it's a blonde woman, but I have no idea. I think I know her, but I can't place her. Or if you artificially excite those neurons, can you just say, oh, I don't know why, I suddenly thought of Jennifer Aniston. Those are the experiments. Right now it's very difficult to do in humans because these are all invasive. It's not so good for the subject, but you can do them in mouse. So all the data, incidentally, all these data are accessible. We built these special purpose APIs as soon as they pass quality control. So we really, once this data passes our internal QC, it goes out. Way before it's available without, you can right now download and get all of our data for free. You don't have to sign anything, no IP. If you are at a drug company, we know a lot of those 50,000 people each month are people working in pharma, great, and we hope you cite us if you make a discovery. And also, interestingly, all this data typically is out there 1 to 3 years before we publish about it, and we've never been scooped. So that's all good. All right. So what we're trying to be about big science, we try to be about team science, and we try to be open science. So the challenge, so we call this our project Mindscope. This is focused on the mouse. We're doing something similar in human, more limited. In the mouse, our project is, that I'll now tell you in more detail, is to describe in great detail the different cell types. It's essential, just like you couldn't make progress in chemistry until at least you understood about A, periodic table, and B, about outer shell valence electrons. We need to understand the different cell types, and we need to understand the specific way in which they interact with each other, their synaptic pathway. We need to do things very quantitatively to get rid of this curse of variability and to better understand it and control it. We need to record, so we want to do what Francis Crick and I, in a paper we published 30 years ago called Brain TV, to record from every neuron and sort of display it on a very big screen so we can now observe at the relevant level, right, the atoms of perception, the atoms of consciousness that I'm very interested in, the atoms of behavior, are neurons, are not columns, are not voxels, but are neurons. And so that's a relevant level that we need to probe. And as I said, with optogenetics, we can now turn these neurons on or off. We need to model it, and we need to finally understand it. And understand it, so we're using a sort of a sense of understanding that comes from people like Herb Simon, David Ma, so multi-level. So we want to understand the brain at the level of the components. What are all the different components at the synaptic level, at the cell type level, at the circuit level? What are the computations that are being performed? And what about the applets, the brain applets, if you want? So there we focus on a very specific one, which relates to attention, object recognition, and decision and memory. So this is the mouse brain and the human brain, photographed on one table. So good order approximations, good number to remember for, I always gave this as qualifying, doing a qualifying examination. A mouse brain is 1 by 1 by 1 centimeter, it's a sugar cube. A human brain, to good first approximate the liter, it's 10 by 10 by 10, or the entire brain. Neurons, there are 71 million neurons in the mouse brain, and there are 86 billion neurons in the human brain, so again it's a factor of 1,000. By mass also, it's like 0.5 gram versus 1,400 gram. Interestingly, if you just go focus on cortex, the neocortex, this layered structure, where all the evidence suggests sort of intelligence resides, and certainly consciousness, there are 14 million cortical neurons and 16 billion cortical neurons in the human. So once again, a factor of 1,000. So what we're getting ready, this is a telescope that Caltech is building. It's a 30-meter with an effective resolution. It has these independent hexagonal mirrors on Mount Kea. And this is a project, it's a big instrument, cost $1 billion. We want to do something similar in sciences to build brain observatories that are highly standardizable, highly reproducible, highly accurate, and scalable. Well, just like in biology, similar to the observer model in astronomy, well, let's say in a couple of years you have an interesting idea and a proposal, and you apply to a selection committee at the Allen Institute to do a particular experiment in olfaction or in memory or whatever. Right? So you've done some prior work, and so you believe that the mouse can do this, and you have a specific hypothesis, and then we implement it in our observatory, we run it, we generate the data. The only proviso is that the data will be made publicly available and accessible. And this should lead to, because it's a reproducible process, we have standard operating procedure, we have engineers and process engineering and technician, it's going to be very reproducible, and the data is available so anybody can look at it. If he doesn't believe you, he can go back and look at the original data. Rather to peer at the origin of space-time in the cosmos, we want to peer at the brain that gives rise to the mind that wanders in the first place, in a creature similar to us. I didn't want the label, because the point I wanted to make here, if I remove the label and I remove the scale bar, so one is roughly 3 times thicker than the other, as I said before, the structure is remarkably similar, whether it's a mouse cubic millimeter or a human cubic millimeter. And indeed, so this is on the left is the hierarchy we get from the monkey, similar in human. On the right, we have a hierarchy for the mouse. So each one of those boxes on the left and on the right contains many thousands or 100,000 of neurons. Each one is a region of the brain. On the left that we've identified with electrodes and in humans with fMRI. On the right that we've identified with neuroanatomy and now also with recording, but at the cellular level of resolution. So let me show you 3 products. So what we're doing now, we're essentially generating 3 things. We're generating detailed connectivity diagrams, making them available. B, we're generating detailed description of the individual neurons, how they behave electrically. We provide models, either sort of statistical models or biophysical models of them. C, we give you the genes expressed in them, so we give you high resolution a description of the individual models to the community. And C, we're building up these brain observatories where we observe under these highly reproducible conditions particular computation the animal has to perform as it runs past various displays and stops or doesn't stop, depending on the task. And then the challenge, both for us, for our in-house modeling team, as well as for the community that we'd love to get any help on, we can get, is to model, right? Is to take the models for the individual neurons together with the known connectivity that we have, take computer model to model the dynamics in order to reproduce the observed behavior. I mean, that's a challenge that we have to do. So I'll show you these 3 products. So one is the detailed connectivity atlas. So here what we do, we essentially inject virus. So again, it uses cool virus molecular biology technology. You inject the virus in a particular type of cell that expresses a fluorescent gene. You wait 2 weeks till it diffuses all the way from the cell body all the way to the synaptic terminals, and then you image it. You take the animal, you sacrifice it, you cut it up very thin slices, and you do high resolution 0.35 micrometer microscopy. It takes roughly a day and generates you almost 750 gigabyte of a terabyte of data in 1 hour. So we have in 1 day. We have many of these instruments. And then we do a lot of informatics processing. You know, we have to stitch it, we have to normalize it, we have to segment it, we have to stitch it together, we have to remove overlap, we have to, control for variation in the laser power across the different images. So there's a lot of informatics that goes on. But finally, we push the data out into a viewer. So now you can see this in a nice 3-dimensional view of the mouse. And we have now a couple of thousand mice where we have this wiring. So it took like 100 FTEs over 4 years. This came out in Nature last year. So this is the basic process. So there we inject, I'll show you a movie. So there you see it's injected in a particular part of the brain called the LGN. There we map it in a standardized brain. The brains, of course, are variable, but if you take enough brains, in this case 1,700, and carefully superimposing using spline and other interpolation tools, you can get a very high resolution, quite accurate atlas. Of course, one very important difference between us and mice are all inbred, right? So these are this is standard laboratory mice. It's been used since, probably since the 1930s or 1940s. So they're highly inbred. They're called C57 black. We always do the same date. They're all male. They're always 65 days old. We always do it on the right hemisphere. Humans, of course, are outbred in biological term. They breed with, they mate with whoever they please. And so you get, of course, much more variability. So this is one advantage of working with a lab animal. And then you can sort of map it into detailed three-dimensional atlas, where it's now registered with the individual pixel, as I mentioned, is 10 micrometer. And so here, this shows you one data set. As I said, you can download download a variety of these different data sets at different levels of resolution. The original one is, of course, I don't have a terabyte of data on here to show. But so you can see, so these are 140 cuts moving from the front of the brain to the back, from the olfactory lobe all the way to cerebellum. And so you can, in principle, if you sort of have, if you know something about the anatomy of the brain and look at it every day, you can really beautifully visualize this. So here it was injected into the visual cortex, and it projects to all the various parts of the brain there. And you can zoom in great detail. As I said, the individual pixels are 0.35 micrometer in X and Y. You can visualize this in 3D. As I said, the viewer is in 3D. So if you do that, this is here done in wild-type animal. And what this is, so here we injected in roughly 500 mice. We injected in a very specific area, in the medulla, in the pons, or in a particular part of the cortex. Which one, you have to zoom in here in high detail. So we injected in this one part of the medulla. Right, we wait 2 weeks, so now all the neurons that are in the medulla, they get infected, and they carry this green fluorescent proteins. And so now you can see where their wires go, and you can see, throughout the entire brain on the same side and on the opposite side of the brain where they project to. And here we use the signal itself is log normal distributed over 5 orders of magnitude. So here we put it in log coordinates. So red means very strong connection, blue means very weak connection. But it's very difficult to binarize because it really goes over this very large range with this log normal distribution. But so essentially there you get a high resolution, detailed atlas of the connectivity. It's still at the population level. In other words, you get all the neurons that sit in layer 5 of this particular part of cortex that project from here down to LGN or down to pons. We're now pushing this atlas even deeper. Ultimately, of course, we want to go to the level of individual neurons. And there, for example, so here you see just in primary visual cortex, so it's a great deal of interest in primary visual cortex, also called V1. Collectively, we as a field know more about this part of the brain than any other part of the brain, certainly any other part of cortex. And this by itself gets input from 42 different areas. So there in the top row, you see all the different areas in cortex and subcortex that project into here. And here you can see the various regions that project out to. And as I said, there's a weight associated with all of these projections. So as I said before, at the neuronal level, fan in, fan out is high, between 10 to the 3 and 10 to the 4, and even at the network level, fan out is quite high. So here you can see distribution strength in these logarithmic representation is roughly normal. You can do, you can do a lot of network sciences, you can do analysis, clustering, coefficient, no degree, and all of that. So using this data, we're now constructing, as I said, a very high-resolution, three-dimensional common coordinate system because, how can you do science if you don't know exactly where you are? So this will enable us to exactly pinpoint every time we record from a neuron in a living animal or in a postmortem tissue where we are with great deal of precision. This is what I mentioned before. The fact is that they're not just cells in the brain, as in any other organ. So a great discovery that was made roughly in 1810 or so was that all biological organisms consist of cells. That was controversial till the end of the 19th century, when it was accepted even for the brain, that the brain, like any other biological organ, consists of many cells. But it doesn't just consist of neurons and glia, supporting neurons, not just out of excitatory neurons, inhibitory neurons, but as I said, overall probably 1,000 different cell types. And here you have them in the retina. So on the top you have the different types of cone. Those are intermediate neurons. In the bottom here you have all the output neurons, the neurons that actually make up the optic fiber, the 1 million cable, the 1 million axon heavy cable that leaves our eyes and goes to the rest of the brain. And whether you're looking in a rabbit or in a salamander or in a mouse or in a monkey and us, it's all very, very similar. So we have this for the retina. In fact, the only place where we have this beautiful cellular description. Each one has a specific function and a very specific morphology. We don't know what those cell types are for the cortex, and so we need to discover them. And so we use different modalities. You can discover them by looking at the electrical behavior at the relevant time scale. We care about perception, and the time scale of perception is hundreds of milliseconds. And so if you record from a neuron, you can see these spikes, and you can put them on an audio monitor. These action potential, we understand quite well how they're generated. You can make models of those. You can look at the morphology. You can look where they project to, and you can look at the genes that they expressed. And then you can do clustering analysis to try to find how many discrete clusters are there. Is it actually true, the hypothesis, that every one neuron falls into one class, as chemical elements do? Or is it maybe possible that there's some are certainly discrete, other maybe continuum? And how dynamic is that? I mean, are those metastable state? We know during lineage, during development, they are certainly metastable. In the adult, are they really stable, or can they change depending on learning and experience? All of these things we don't know. So we know that we have this pipeline set up where, again, we have technicians that take these animals under standard condition and sacrifice them, take out the same piece of the brain again and again, and then record day in, day out from this. And so we have a standard protocol that we use to characterize a cell in this somewhat artificial condition called a brain slice, where we inject current steps and ramps and white noise and pink noise and all sorts of things to characterize each cell under standard protocol. Because then what we can do, we can build models then. We can either make from the electrophysiology together with their shape, I'll show you that in a second, we can make models. We can either make point models, sort of generalized linear models or generalized linear integrated fire models that are very simple with 4 or 5 degrees of freedom, or we can put in more bells and whistles, whether you have adaptation, where you have time-dependent adaptation, where you have time and voltage-dependent adaptation that have maybe 100 degrees of freedom, to a much more complicated but much more realistic biophysical model that actually model the physical reality of them. The point model, the advantage is they're simple, they're relatively abstract, and most of them we have a convex function, so we can derive the optimal solution. For those, we need to use genetic algorithms or other tricks. So we have, as I mentioned, we have automatic fitting procedure that might explain 70 or 80% of the variance in a brain slice. Now the bad news is a brain slice, it's actually a comatose brain. Because what you do in a brain slice, and we do it for human brain as well, you take out a piece of brain and then you put it in a dish, and now all the neurons typically tend to be quiet. And of course, in a normal brain, neurons are never quiet. Even when you're in a deep sleep, there's always electrical activity going on, which changes the character. But you know, it does allow you to very stably record and characterize these neurons. So you can do these statistical models. Then, because we also get the shape of the dendritic tree, we can use that together with the electrical property to derive these very detailed biophysical model. So here you see one, for example, on the right, it's just generating action potential. This is using a standard computer model called Neuron that models the partial differential equation describing diffusion and electrical propagation along these cable structures. And we can do that automatically now, or semi-automatically now, for a variety of neurons. So here, on the left, you see data in response to current injection. Here in the middle, you see the best model for this. And here you superimpose the individual action potential. That's the real and the simulated one, and here the various shapes. So the first data set release with all of this will be out in 3 months from now, and then we continue. We're now really accelerating. So we hope each year we can do a couple of thousand of these cells and make them available to all the modelers out there. And then, for example, you can see to what extent they generalize. So you can do leave out stimuli. So here the black is when you stimulate, the same neurons are noisy, right? So experimentally you use the same frozen noise and inject it again and again 4 times, and you can see the different spike patterns you get there. Here's a different voltage pattern. And then you can see to what extent do your previous model that were trained on current steps generalize. So you can do a lot of stuff there. Okay, so if we can do that pretty well, the drawback is two. One is, as I said, we can describe neurons, the electrical behavior of neurons in a comatose brain. B, everything we do from the point of view of the cell body. And we know there's a lot of cool stuff going out there, a lot of computational nonlinearity going out in the dendrite to which we're relatively blind to. At the same time, because we're trying to understand how many different types of neurons there are, we do what's called transcriptomics, where you essentially go in and look at the RNA of individual cells. We do it at the single cell level. We do very complicated procedures both in human and in mouse brain. And then you do clustering algorithms like iterative PCAs, ICAs, all of those things to, for example, arrive that we see in this particular part of the brain that we study, primary visual cortex, we see 22 different cell types that are pretty unique based on purely molecular characterization of the genes. 9 excitatory, 9 inhibitory, and 4 sort of clear non-neuronal ones. And here there are very interesting patterns for the experts. Some are very discrete, and others they're sort of a continuum, which is really very interesting. And then lastly, what we're doing with recovering the shape. And this continues to be a big, big technology hurdle. Because so here we have a neuron that we infected in a living animal now. So you take a living animal, you take one plasmid of a rabies virus. It's a modified rabies virus, but it's related to the one that if you get bitten by a rabid dog, what happens? This rabies is pretty clever because it latches onto you and travels into the neurons and travels back up through the synapses into the previous neuron and then travels back up to the previous neurons until it reaches to your brain and makes you do bad things that ultimately will lead to you spreading the virus to some other brain. Now, biotech has hitchhiked that and done the same where you inject into one neuron and that can go backwards and infect other neurons, and we couple it with a green fluorescent protein so we can light it up, which is pretty cool. Now we have computer algorithms, VAS3D in particular, that can reconstruct these dendrite. That we can do almost automatically if we have enough signal to noise. The problem is the axons, is the output wire. So again, a few numbers to remember. Good for any qualifying exam. In a cubic millimeter of brain, there are between 50 and 80,000 neurons. Okay, 1 cubic millimeter of brain, 50, 80,000 neurons. There are roughly 2.4 kilometers of axons, and there are 200 to 300 meters of dendrite in that 1 cubic millimeter, and there are a billion synapses, 10 to the 9 synapses in the cubic millimeter. So essentially, think of it a crystalline-like array with synapse every 1 to 1.1 micrometer, space constant. The axons are very, very thin. They can be up to 0.1, so below light level resolution, and they go everywhere. And so here we have a problem. If you digitize them, try to and reconstruct them, you get these gigantic files, 55 gigabytes for one axon of one neuron in the animal. And that's very laborious to do. So this still takes right now 400 hours. So we're working on automated techniques to do this much faster. And we have a large-scale collaboration with Google to do both this as well as EM reconstruction automatically, so we can do all of this with many, many fewer people. So what you get things like this. It's a single neuron, a layer 5 pyramidal cell, for those of you who are expert, that sits in primary visual cortex and then sends its axon way, 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 way down there, all the way to the thalamus, to the colliculus, and down to the pons. So unfortunately, this sort of data we only have very rarely. There are very few people, very few labs that do this, and nobody does it systematically at large scale just because it's so laborious. So we're working closely with machine learning people on this to try to greatly accelerate this because we want this for all the neurons. We're also doing this for humans. So we now work with 5 different neurosurgeons in Seattle. So typically what will happen here in the setting, either epileptic seizure or tumor surgery. So let's say you have an epileptic seizure that originates from your hippocampus. So the area highlighted there under the yellow arrow. So the doctor, because drugs don't work anymore, we know by and large if you can locate the seizure, the focus of the seizure, to then you remove it, you may be seizure-free for the rest of your life. Now, to access that hippocampus, you have to go from the outside, and so essentially the doctor has to tunnel his way into there, and he removes that piece of brain in the red box, like over there, right? Looking like sushi. So typically, it may be this big. Sometimes it may be this big, just depending on, it's roughly always from the same location. So we stand outside the OR, we wait for it, we put it in a special glass that bubble carbon dioxide and oxygen. We rapidly move it to the lab because we're now recording from it. You got to remember, 30 minutes ago, this was part of a living brain and may contain the memory of her first kiss in that part of the brain. It's a human brain after all, not a monkey brain anymore. Although a mouse brain, the neurons look fairly similar. In fact, they're bigger. So in general, the bigger the animal, the bigger the neurons. And so we find it's relatively easier to record from. And in fact, we can record from much longer than from a mouse. The brain seems to be much more resistant to oxidative damage. So we can get these beautiful electrical recordings from neurons. So we have this program there as well, where we're trying to do the same characterization of the morphology and the electrical behavior together with computer models and together with the genes that are expressed there to make that available to do clustering. Okay, lastly, we want to build these brain observatories. So this is a mouse, just for fun. One of the investigators trained it to recognize, to stop at pictures of me and to run past pictures of the other investigators at the institute. All right, now what you don't see, it's pretty good. What you don't see is, normally we would put when the mouse does this, we swing a microscope in place because we have a team of 4 to 6 neurosurgeons, mouse neurosurgeons. They operate these glass windows into their skull, and then they wait 2 weeks for the mice to recover, and then each day the mice will run for an hour or two, and we show them different stimuli. Typically, we show them gratings or various objects that we define, that they've been trained to learn, to recognize, and then they have to perform simple tasks, like stopping, like here. You stop at one particular shape, but you don't stop at another, and so you have a behavioral readout. You can see, do they detect it, do they not detect it, do they make mistakes? You can compute false positive, false negatives, etc. You can run ROC curves. While you're doing all of that, and so you can see the mouse gets rewarded for correct behavior through that snout, gets some water. While you're doing all of that, you're peering inside the brain at cellular level resolution, right? That's a beauty. We can observe, we can do something similar to fMRI through the skull. We can see so-called wide field imaging, where we can observe the large-scale behavior of the neuron, but even better, we can observe individual neurons. All right, so I told you already about this. So one advantage of the mouse is its brain is not only much smaller, but it's also smooth. Because we have this big brain that needs to fit into the skull, we have this heavily convolved. Mouse is a smooth brain, so with a single observational window of 5 millimeter, you can observe primary visual cortex and all the bevy of surrounding visual areas with one observational window. It's another advantage of working with the mice. And what you see here is one of the output of these observatories, called so-called 2-photon calcium imaging of GCAM6 activity. So what this is, we have a microscope, a 2-photon microscope, where we can record the optical signals in an engineered mice, either an engineered mice or with a viral injection, where the virus or the engineered mice carries a protein that converts its electrical activity, its spikes, into a signal, into a calcium signal that we can observe, that we can pick up here. Right? So here you see, in a living animal doing a particular task, you can see part of the network as it switches. So as the mouse is thinking, do I need to run past this, or can I stop and get my reward? This is, we can observe the individual components switching, as it were. So right now we're limited, recording maybe 300 to 400 of these, but we're doing this in a highly reproducible way, so we can go back and record from different layers using different genetic encoded of these molecules, so we can overall record the entire activity to some standard data set across the different parts of the brain. And then do a lot of, you have to, of course, understand that then there's already a lot of processing done here, but we have to do further processing, because what we essentially want to extract is, we have to do region of interest, and then detrend it, etc., and then essentially get a time course from all these different neurons that corresponds to the time scale activity, to the time scale of activity of these neurons, together with the label and all the metadata, and make that accessible so we can analyze it. And so for that, we have to track the overall brain state that we can do here using this wide field imaging. When a single trial, we can image through the skull almost the entire brain activity. We need to keep track of its eyes. We need to keep track of its actual behavior there, and then we need to map it, all that activity, to these detailed internal functional maps. These are similar to the fMRI maps in humans, except we get, of course, with much higher resolution and at the cellular level. And so we need to keep track of all that activity. So we've had the challenge over the last 6 years to move these pipelines. So pipeline we call a large structured set of people and instruments from, where we put in, let's say, something, let's say a mouse, and out we get out some product like the genes expressed, particular genes that are expressed in this mouse, or its neuron activity. And so we've shifted progressively from using postmortem tissue and doing pipeline on that to using a living tissue, brain slice tissue, where we record from, to the most difficult one that we're now trying to launch by March of this year, where we record under highly standardized condition in this living animal. Okay, so we have these three large-scale atlases for the mouse: the detailed connectivity atlas, this detailed single-cell characterization atlas, the detailed cortical activity atlas, and they all use same type of mice, same age, same hemisphere. We map them all within a common coordinate system. Never mind, that's for the experts. And it all goes into centralized, standardized database, and then is pushed out there to the community. Since what we want to do, both for us as well as for the community, is to challenge our own modelers, theoreticians, as well as there are many, of course, now in the community of computational neuroscience, where we show them the input, what is it that the mouse saw, visual input, where we show them the different cell types, where we give them the connectivity, where we give them the observed behavior, where we then want to use, as I said, computer models that take the structural data about connectivity and about neurons to try to reproduce the functional data, and thereby to gain a better understanding of the computation that's going on in the mouse. We did this last year, but we're going to do this year. Next 5 years, for anybody who's interested, we're doing this workshop on Friday Harbor, which is this beautiful island north of Seattle, in the Fuca Strait between Canada and Washington. In the summer, 2-week-long workshop, big data and data analytics on these data sets. So we're looking to reach out in the community to do that. And we have these detailed milestones. As I said, unlike a university, we're on a tight timeline here. Right now, of course, we are early 2015 to try to execute all those things over the next 8 years with this team. So let me make some concluding slides. So this is a large-scale effort, large-scale not by physics but by biology. It's a 10-year effort under one roof, whose fruits are freely shared with the public. They come with a few challenges. So we have to focus on a few large-scale projects. Unfortunately, what I have to do now, half or more of my time, I have to say no to internal people that want to collaborate with external people, to people who want to fund us, to outside collaboration, because as we all know, you can only do a few things very well, and so we choose to focus on a few large-scale projects that we can do. It requires a tight integration between the different teams, which is easy to say. It's very difficult to practice, because you have people who've been educated sort of with different views, different standards, different coordinate system, all of that. And even in the same building, even if they're in the same management structure, it's not easy to get them to work together. It's hard work. And of course, like any other team science, there's this conflict, because we need to harness the creativity and drive of individual, but emphasizing and rewarding the team aspect. So I've really learned the need from Paul, the need for deadlines, giving smart people a lot of money and just say, well, produce something, think deep, think really deep, and I give you a lot of money and solve all the world's problem. Traditionally, that hasn't worked out too well. And that's also been our experience. So I think it's a great management tool, in fact, that we use it as such, that we have milestones, etc., which we have to accomplish. So one can also think of it as an experiment in the sociology of neuroscience. And this is half of our team in Seattle. Now let me end. I have a cool movie I wanted to show you. It's beautiful for an ending, just to show you the complexity of the brain. And I think, in fact, there's a theory called integrated information theory that posits specifically out of this complexity arises our conscious awareness. And here I'll show you the complexity from Stephen Smith, who was at Stanford for many years, who recently also joined us. What he did, so remember, this shows you a piece of cortex, of mouse cortex, with some of the neurons stained, the cell bodies and the axons. So what he did, he has a particular technology called the ray tomography, where he takes this little sliver of mouse cortex, which is from top to bottom, maybe here, this is a millimeter across or so, maybe a millimeter and a half. And this probably is a little bit, this I think is 100 micrometer. Now a hair, typically human hair is between 50 and 80 micrometers, so it's very thin. And it's only, this slide movie you see is only 70 micrometer thin, and shows you with different antibody staining 3 structures. So in green, you see a green fluorescent, you see one set of neurons, particularly the dendrites of these layer 5 neurons. In purple, you see the synapses, and 95% of all the synapses are visualized here. And I believe in blue, you see the axons. And this is, compared to the fMRI picture that many of you will be familiar with, of course it's tiny, right? So this entire movie you're going to see takes place in a tiny, tiny voxel. So the typical fMRI voxel would go from the top of this room roughly to the bottom. One voxel, and of course typically you see 3 by 3 by 3 or something. So just to show you the complexity of it all. Let's see. Wait, there was a movie. There. It's part of a cortex, somatosensory cortex. So we're now going to zoom in on this piece. Millimeter by a couple of 100 by 70 micrometer. We start at the top, layer 1, top of gray matter. This is essentially in human, it's called gray matter. So green are the top dendrites that branch out on the top. These are all the synapses. Blue are the axons. Synaptic input from the outside comes. of the cell bodies. This is the most complex piece of excitable matter in the universe, and this gives rise to our consciousness. Thank you very much.
Fascinating talk. I think we have a question here quickly.
Yeah, I mean, do you think these kind of projects would lead to what could be classification of a normal brain versus, say, other kinds of brains and maybe explanation of schizophrenia, autism, and
Yeah, specific the connectivity. So schizophrenia is often thought as a circuit disease rather than a single molecular disease. And so if you know what the normal brain is, some standard, of course there's going to be a probability distribution, you should be in a much better position to judge what is normal and what's pathological. Right now, this level of resolution you can only obtain in a post-mortem brain, which is not so good for routine clinical scanning. But you know, science advances constantly.
Sergio.
Yeah, so this audience, one of the problems that occupies our work is the problem of interference in wireless communications. Now here you have dendrites and axons that are very highly packed, and the question is whether there is any interference among their communications.
It's a very good question. So I looked at that quite a bit. So there's something known as the ephaptic effect. Right, so if you have a bunch of cables, of dendrites next to each other, and one generates an electric field, you will get coupling. Typically, the coupling seems to be very small. So the hypothesis of most is, except in pathological conditions such as epileptic seizure, when everybody fires, when you probably do get volume conduction. In general, volume conduction, i.e. interference, is probably not relevant in on a normal condition. That's a good work. I think it's a reasonable working hypothesis. It's also born out of the necessity. We can't even understand one of them. How are we going to deal with them interfering? But that's right now the working assumption. Which
Now, the axons have communication in both ways, simultaneously?
No. So the axons typically, again, under normal conditions, will only conduct from the cell body to the output end. You can make them fire the other way, but they don't do that under normal conditions.
Toby, last question.
I may be repeating things that were said in that direction without amplification that I couldn't hear. But as you know, you're talking to an audience, the vast majority of whom are information theorists of one sort or another.
Yes.
I didn't hear the term information in the lecture, and I wonder if you want to say anything that can be said at this point about where you are in understanding information in your work.
Well, so people use information, particularly Shannon type of information, since the early days. Shannon himself tried to apply to into the brain. It remains to be seen to what extent. So, of course, we all believe the brain is the most powerful computer around. It remains to be seen to what extent the computational information theoretical paradigm will prove to be useful. So people compute, they estimate, bit rates, they estimate entropy, they do matching entropy, they do all sorts of things. It still remains unclear to what extent it's a useful metaphor. And it's also a slightly misleading metaphor because in Shannon, of course, you have a sender and a receiver, and one always has to ask who's the receiver in the brain. But it remains a very popular, I used to have an entire class at Caltech on information processing in the brain. So it suffuses everything that people do.
I wish we had more time, but I think we've run over. Thank you again for